You wake up hungry with no alarm, because the people who came before you figured out how to turn grass into breakfast. You wake up hungry with no alarm. Your hand reaches for a warm bowl. That bowl began as grass seed or a root pulled from the ground. This matters now because some modern paleostories erase plants. Conservative, environment-specific estimates put plant shares at roughly 36 to 55% of calories in many temperate, tropical, and desert contexts. This video pulls 5 kinds of evidence and connects them. Tooth isotopes, plant residues, AME1 genes, ancient mouth microbes, and experiments on grinding and cooking. If plants were important, when and how did people make them dependable? That question stays open until scene S11. Start with deep time. Stable carbon isotopes in tooth enamel record the balance of plant types someone ate. The chemistry separates C3 from C4 plants. Many grasses and sedges are C4, so a C4 signal points to grass-like foods or underground parts eaten often. The isotopes show a major shift. Between about 3.4 and 4.8 million years ago, tooth chemistry records more C4 plant input. Fannin and colleagues argue this change predates dental shape change by roughly 700,000 years. In other words, behavior-led anatomy. So tools or routines must have made grass-like foods useful first. Genes and mouth microbes remember diet too. AME1 is the gene for salivary amylase, the enzyme that starts starch digestion in the mouth. Populations with long histories of high-starch diets tend to have more AME1 copies. Ancient dental calculus also preserves streptococcal strains linked to starch digestion. Both human DNA and oral microbes carry a starch signal across time. Archaeology gives direct traces of processing. Starch granules, phytoliths, and charred plant fragments appear in dental calculus and on tools. Grinding stones show we're consistent with seed or tuber processing. These are not one-off scraps. They are the material traces of a practiced technology. Ethnography and dietary ecology set expectations. Across many environments, plants commonly supplied a large share of hunter-gatherer calories. Florin and colleagues estimate plant contributions around 36 to 55% in many temperate, tropical, and desert settings, with lower plant reliance in tundra or coniferous forests. Raw grass seed and many tubers are not easy calories. Starch sits behind cell walls and often tastes bitter. The trick is processing. Grinding breaks cell walls into powder. Heat gelatinizes starch so enzymes can access it. Experiments by Carmody and Wrangham and residue work by Revidin show grinding plus cooking raises net energy enough to make marginal plants reliable staples. Who ran the grinding stones and the hearths at camp? That question links diet to routines and opens S11's practical answer. A concrete case is Ohalo II on the Sea of Galilee. About 23,000 calibrated years ago, one occupation floor produced roughly 60,000 plant remains, about 1/3 of the site's total. The floor had 606 wild barley grains, grinding stones with starch residues, and glossed flint that looked like sickle inserts. Is Ohalo a one-off hotspot or a window into a wider pattern? That question stays open for later. So how did marginal plants become dependable staples? Repeated technology and social practice did the work. People harvested wild cereals with cutting tools and sickle-like inserts. They ground seeds into meal on stones. They cooked the meal over hearths. Grinding opens the cells that hide starch. Heating gelatinizes starch so enzymes like salivary amylase can reach sugars. That harvest-cut-grind-cook sequence shows up in Ohalo II, in upper Paleolithic grindstones, and in cooking experiments. Together, these steps transform marginal plants into stored, predictable calories. Important caveats matter. Plant tissues decay and often vanish from sites. Starch residues can be modern contaminants if protocols are weak. Isotopes separate C3 and C4 input, but cannot be converted into exact calorie percentages without strong assumptions. These limits are real, repeatedly discussed in methods reviews, and they narrow what can be claimed precisely. The convergence matters more than any single line. Isotopes point to early plant shifts. Residues in ground stone where show deliberate processing. AME1 and oral microbes preserve long-term starch exposure. Cooking experiments show big energy gains. When these separate threads meet, the coherent picture is that starch was a shaped, often dependable resource across many places and long stretches of time. Answering a channel thread about night watches, predictable, storable starch made rotating schedules possible. When food can be stored as meal or dried tuber, camps can plan who eats, when, and who takes a watch. If calories are unpredictable, communities are more likely to keep specialist sentries. So diet helped shape who woke at night and how care duties rotated. Big open questions still matter for daily life. Exactly when and where AMI1 copy number increases occurred across populations is not fully pinned down. How widespread was habitual starch cooking across regions and hominin species? Will stricter contamination protocols remove early claims? New work will refine timing and spread, but the overall convergence of isotopes, residues, genes, and experiments keeps the central claim robust. Return to the bowl. That warm porridge is not a late invention. It is a technology built from stones, fire, and practice that shows up in teeth, on tools, and in genes. It shaped mouths, microbes, and social routines from weaning to watches. When you next wake up hungry with no alarm, remember, the people who came before you turned grass into breakfast by learning how to make it reliable. If this rewrites paleo for you, watch the next video. Did storage pits and hearth arrangements enable the first semi-permanent camps? That episode compares hearth floors and storage features across 15 sites.